Okay, what if I told you we could send a message directly to a cancer cell? A message that tells it to just self-destruct? Well, today we're diving into some absolutely groundbreaking research that does pretty much exactly that. Scientists have managed to combine the power of mRNA and CRISPR to build a programmable therapy that takes aim at one of the toughest forms of lung cancer out there.
So yeah, let's just kick things off with that massive question. I mean, think about it. Instead of the usual approach, poisoning cancer with chemo or hitting it with radiation, what if we could just slip a note directly to the cancer cells? A note that basically says, your time is up, self-destruct now. It really does sound like something out of a sci-fi movie, but as you're about to see, it's becoming very, very real.
And you know what? I'm not even going to make you wait. Let's just jump right to the end result. Take a look at this. This is a real microscope image of a lung tumor. See all those dark brown spots? Those are individual cancer cells that have been triggered to undergo apoptosis, which is just the scientific term for a programmed self-destruct sequence. This is what happened after the treatment we're talking about today. The only question is, how did they pull it off? Let's get into it. All right, so to really get how the solution works, we've got to understand the problem. And the villain of our story is a tiny, broken switch deep inside the cancer cells, a switch that, for decades, has been almost impossible to turn off. That villain has a name. It's a gene mutation called KRAS. It's a huge problem, driving about a quarter of all non-small cell lung cancers. Now, a normal KRAS gene is like a switch for cell growth. It turns on when it's time to divide, and then, crucially, it turns off. But the mutated version, it's like that switch got jammed and is now permanently stuck in the on position. And that, right there, is what leads to the runaway cell growth that we call cancer. So how do you fix a switch that's stuck on? Well, for a long time, the only idea was to use drugs to kind of jam a wrench in the works, to block the protein. And that can work, for a bit. But cancer is sneaky, and it almost always finds a way around the blockage. But what if you didn't have to just block it? What if you could just go in and cut the wires to the broken switch entirely? That's CRISPR-Cas9. It's not a blocker. It's literally like a pair of molecular scissors that goes straight to the DNA, the source code, and just snips the faulty gene, shutting it down for good. All right, so this all sounds amazing, right? We've got these incredible molecular scissors. But, and this is a huge but, how in the world do you get them inside the right cells in the right organ? I mean, having a powerful tool is great, but if you can't get it to the job site, it's useless. It's a classic delivery problem. Yeah, the most common delivery vehicles for things like mRNA are these little guys called lipid nanoparticles, or LNPs. You've probably heard about them from the COVID vaccines. But here's the problem. For whatever reason, standard LNPs are naturally drawn to the liver. It's like an Amazon truck whose GPS is permanently set to one single city, and that city is the liver. If your package needs to go to the lungs to treat lung cancer, you're out of luck. You need a whole new delivery truck. So that's exactly what these researchers did. They set out to build a new delivery truck from scratch. They tinkered with a whole library of LNP recipes, changing up the ingredients to find a combination that would specifically target lung cells. And wow, look at the results. That bar on the left is the standard liver-loving LNP. Now look at A6 and A8. They delivered their cargo nearly 10 times more effectively. I mean, look at that jump. We're not talking about a tiny improvement here. This is a game changer. They found their secret sauce. And here, thanks to a super powerful electron microscope, we can actually see why they work so well. These aren't just plain old spheres. Look at A6 on the left. It's got this, well, it's like a little bleb, an extra bubble on its side that seems to help it do its job. And A8 on the right has this cool, complex, multi-layered structure. It's this unique, custom-engineered architecture that lets them slip past the body's defenses and get right into those lung cells. Okay, so they designed this beautiful, custom-built delivery system. The engineering is brilliant, but does it actually do the job? Does it get the CRISPR scissors where they need to go? And more importantly, do those scissors actually cut the bad gene and kill the cancer? Let's look at the evidence.
So first things first, does it even work in a petri dish? The team took their two best nanoparticles, A6 and A8, and used them to treat human lung cancer cells with that faulty KRAS gene. Now just look at the bar for A8 1-to-1. That is incredible. They are successfully editing the target gene in almost 90% of the cancer cells. That is a ridiculously high hit rate.
Yeah, let that sink in for a second. 90%.
To get that kind of precision with a non-viral system, which is basically a tiny engineered bubble of fat carrying this complex genetic message, that is just a phenomenal result. It's proof positive that their custom delivery vehicle is hitting its mark. Okay, so editing the gene is cool and all, but that's not the ultimate goal, right? The goal is to kill the cancer. And this data right here, it screams yes. What you're seeing is a measurement of apoptosis, that programmed cell death. And the bottom line is that the A8 nanoparticle caused a 3.6-fold increase in cancer cells dying off. So the scissors got delivered, they cut the gene, and the cancer cells got the message to self-destruct. So it works in a lab dish. That's fantastic. But, the real world is messy. The human body is way, way more complicated. So now we get to the ultimate test. Can this system work in vivo, inside a living, breathing organism? This image shows the challenge. A solid tumor growing inside actual lung tissue in a mouse model. That's the real target. And boom, here it is, the proof. This is the very same image I showed you at the beginning. After they treated the mice with their A8 nanoparticle, they used a special stain that lights up any cell that's in the process of self-destructing, and look at it. The tumor is just packed with those dark brown spots. It worked. The special delivery trucks found the tumor, dropped off their package, and the self-destruct sequence was initiated. And we're not just looking at a pretty picture. The numbers back it up big time. When they actually counted these self-destructing cells, the group treated with the A8 formulation showed a 3.5-fold increase over the control group. This is the hard data that says, yes, what you're seeing is real, it's significant, and it's working right where it needs to.
So let's just take a step back for a second. What does all this really mean? Because this is about so much more than just a new way to fight lung cancer. This is a peek into the future, a proof of concept for something much bigger. So let's just break it down one last time. Super simple. Step 1. Scientists engineer a custom LNP, a biological delivery truck that's programmed to go to the lungs. Step 2. That truck carries a precious cargo, mRNA instructions for building CRISPR. Step 3. Once inside, CRISPR acts like molecular scissors, finding and breaking the mutated KRAS gene. And step 4, with that broken on switch finally disabled, the cancer cell gets the signal to self-destruct. You don't even have to take my word for it. The researchers who did this incredible work said it best themselves in their paper. This is a powerful proof of concept. They've shown that this kind of non-invasive strategy has very real potential for a type of cancer that has always been incredibly hard to treat. They've written a whole new playbook. And that really leaves us with this huge, exciting question for the future. Think about what we just saw. A custom delivery vehicle taking a specific genetic instruction to a specific place in the body to fix a specific problem. What if we could do that for other diseases? What if we could create personalized nanoparticles to go to the brain or the heart or the pancreas? This isn't just about a new weapon against lung cancer. It's a blueprint for the future of programmable medicine. A future where we might be able to treat all sorts of genetic diseases just by changing the message inside the package.